Ariel Gomez, Vice Chair of the Council on Hypertension. And it's my great pleasure to be here with Nobel laureate, Dr. Gregg Semenza, who just gave a fantastic, illuminating keynote lecture on hypoxia-inducible factors in physiology and medicine. Dr. Semenza is a professor of genetics medicine and director of the vascular program at Johns Hopkins. And he received the 2019 Nobel Prize in Physiology or Medicine for his landmark discovery of HIFs. So, Dr. Semenza, thank you for enriching us with your contributions to the scientific sessions of our council and for agreeing to this brief interview.
Thank you, Dr. Gomez.
Sure. I have a few questions, and the first one, I think, is an easy, very easy one, and is, what were your beginnings? How did you start thinking about science? You know, we are trying to ask a few more humane kind of things for the trainees, and so to see that you're a human, not just a Nobel Prize.
Well, you know, I was interested in genetics during high school. Then in college, I, you know, became interested in medical genetics. And at that time decided I would go to medical school, get an MD and a PhD, take care of patients with genetic disorders and do genetic research. So that's how I got into this. And then, well, you know, we have a genetic disorder that's associated with the HIFs that was very fortuitous, but we've also spent a lot of time, you know, studying very common diseases like cardiovascular disease and cancer. So we've just tried to follow the science, and at every stage ask, you know, how can we potentially target this therapeutically? So in the last you know 30 years, we've been able to learn a lot in terms of the basic science, and now we hope to be able to apply that to different disease states.
Sure. We'll get a little more to that in a second, but was there a source of inspiration in your early stages of your career?
Yeah, so I had a high school biology teacher who really inspired me. Her name was Rose Nelson, and she was a high school biology teacher, but she had a PhD and had done a postdoc at Woods Hole, and she really understood the research process. And she would describe to us the discoveries that were made in biology, and rather than just reciting dry facts about, you know, the leg bone is connected to the foot bone. And so she really transmitted the excitement of scientific research and discovery. And that really inspired me to a career in biomedical research.
That's great. So how easy or difficult were your early steps in research?
Well, yes, my life as a graduate student was a very checkered one. So I was in a lab for about a year and really didn't make much progress, and just didn't see myself making a lot of progress. And so I took the kind of traumatic step of changing to a different lab and starting all over. And I was in a lab that was studying the molecular basis for beta thalassemia in a particular family. And when I sequenced the beta globin gene, I found the same mutation that had been found in the family before this one, which suggested that there had been some sort of contamination. And so I was forced to start all over again. And at that point, you know, I was very depressed. And so I went to see a psychiatrist who happened to be the father of two of my classmates in med school. And I told him what had happened. And he said, well, you know, Greg, it sounds like you have good reason to be depressed. And so I don't know, that made me feel much better. I went back and started over again and was able, to finish the work in time to graduate with an MD PhD in 6 years. And the work ended up being published in Cell. So yeah, when I was in college and worked at Boston Children's Hospital doing research, my mentor, Park Gerald, used to say, search and research. And if you're not kind of willing to confront obstacles and see them as challenges, then research is probably not for you, because that's part of the excitement of it, is overcoming different types of obstacles to move the work forward.
Yeah, I'm glad that you just brought that up, because my next question was going to be, what were your roadblocks? But you just explained it in such a great way, and I think for the junior investigators, this is a great example of resilience and determination, to continue going, and that not everything is a field of roses, right?
That's for sure. And when we were trying to clone HIF1, it was also a similar situation where we had done an experiment that suggested that there was only one subunit, and so we thought we could identify the cDNA by looking at different bacteriophage that express human genes, and then using the DNA oligonucleotide binding site as a probe to identify a colony that expressed the human cDNA. And we screened millions and millions and millions of bacteriophage. We got negative results. And, at that point, I had to decide what to do next, right? Should we just give up and let somebody else do it? That just didn't seem like a very good idea. Or should we try a completely different approach, which was the biochemical purification of the protein? And at that time in my lab, we didn't even own a fraction collector. It was a molecular biology laboratory. But fortunately, Tom Kelly's lab was across the street, and his lab was one of the first labs to purify a protein based on its binding to DNA. And so we got a lot of help from his lab in performing the biochemical purification of HIF1. And we got just enough protein to get a little bit of sequence, and then design degenerate oligonucleotides based on the amino acid sequences that we had gotten. And we were able to identify cDNA encoding the HIF1 alpha and HIF1 beta subunits.
Wow, that's amazing. I mean, that's an enormous amount of work.
As you could tell from my presentation, we have many collaborations with labs both at Hopkins and elsewhere that really help us, move the work forward, because we can take our complementary skills with the skills of the other lab that may be focused on particular disease models, for example, and together, come up with interesting results about the role of HIFs in different disease processes, without becoming experts in any one of these things. And so that's been a very enjoyable part of the process, because everybody looks at research from a different point of view, uses different approaches, and to collaborate with different labs that take different views of the same process, I think is very valuable. And of course, the opportunity to really develop lifelong collaborations. So we've collaborated with Joe Prakal's lab for over 25 years, and Andrei Prabakar's lab for over 20 years. So it's one of the many exciting and enjoyable things about doing biomedical research.
Yeah, perhaps research is one of the most democratic activities, right, that we have.
Well, not only that, I mean, I think that international leaders could learn something from the way that scientists collaborate around the world, right?
It's so good. Yeah, I agree, totally agree. So good to be able to have all these many collaborators, and as you said, it's so enjoyable to be able to learn from them and so forth. I wanted to ask you: was there a moment in your career? I mean, when I know that about the roadblocks, but when you realized that you were on the right track, the sort of aha moment. If you remember that, I don't know.
Yeah, well, there were several aha moments like that, and you know, one was to first identify the HIF1 by virtue of its binding to the DNA fragment, and this was another real make or break point, because DNA interacts with proteins in a very idiosyncratic way. So one protein may need different salt concentrations. You may have to put different carrier DNA to block nonspecific binding. And if you're looking for a protein to bind to a DNA fragment, you don't know what those properties are. And so all you can do is search for binding and hope you find it. And if you find any slight degree of binding, then you try to optimize the conditions. And so my postdoc, he was doing experiments every day in the cold room, trying different binding conditions and looking for binding of nuclear factors that were present in hypoxic cells that make EPO and weren't present in cells that were cultured under high oxygen conditions. And every day he got a negative result. And he had a stack of autoradiograms on his desk that was about this high. And one day I was just flipping them, looking at them, just holding them up to the fluorescent light on the ceiling. And after, going through half the pile, there was this one autorad with a very, very faint band. And I said, "Did you see this? Did you see this?" And of course, he didn't see it because it was in the middle of a pile of failed experiments, right? But that turned out to be HIF1. And we then, were able to optimize the conditions. And in several weeks, he had very, very strong bands showing binding of a factor to this DNA sequence. And then, as I said, that enabled us to then purify the protein. So there's really something to be said for closely going over the primary data. Yeah. And I think that as we go on in our careers, things tend to get delegated and filtered, and we only see the good results, right? Mm-hmm. And when that happens, you may miss something that's important because the trainees who are doing these experiments may not understand what is the good result or an important result. And so, yeah, I think as much as you can, it's important to look at the primary data and
exactly.
Make sure you don't miss anything.
You are teaching us already a lot of things. I just want to ask you, so at some point you became a pediatrician. I'm a pediatrician too. I'm a pediatric nephrologist. So of course I'm interested in EPO. Fortunately, we have EPO now, recombinant. So the study of medicine, I guess, influence, maybe I'm asking actually, your research in some way, right? Did it provide you with a breadth of knowledge that then allow you to begin to focus on those fundamental questions?
I was trained as a medical geneticist and attended on the medical genetics service here at Johns Hopkins for 15 years or so. And then focused after that on, the research and teaching. But of course, our research remained very, very close to specific disease processes. And of course, my training gave me a perspective about that. about the need for treatments for various disease conditions, and so you know, as we've gone on, we've gone on with the goal of trying to, you know, develop new therapies that could be useful for the treatment of various diseases. At the moment, we're in my lab, we're focusing on HIF inhibitors as a way of blocking cancer progression, and also different disorders associated with ocular neovascularization. And then, of course, I mentioned that there are many groups that have focused on trying to induce HIF activity by inhibiting the prolyl hydroxylases. And so for all of these, you know, the thing is that HIF1, as I showed, does many things. And the question is, you know, for any indication, is there a therapeutic window, right? Can you achieve the desired therapeutic effect without untoward side effects? And of course, you know, we know the answer to that can only be gained by clinical trials, because however good our models are, they're not good enough to answer that.
Right.
And so, as I illustrated with the anemia trials,
Exactly.
you know, it appears that there are side effects of the drugs that may preclude their use, at least as they were used in the trials. And this really didn't come as a surprise, because the people with the erythrocytosis that I described that was due to VHL mutations, those individuals, many of them die in their 50s with arterial and venous thrombosis,
Right.
which we don't fully understand. But it clearly says that if you have increased activity of the system, this is a potential pathogenic effect. And so to see similar things, not as great in terms of mortality, but yet to see a signal of that side effect in the drug treatment was very interesting. And obviously it was unfortunate and unwanted, but not totally unexpected.
So I just the last question, so we can give you a break from all these activities. But you made a fundamental discovery, really, how we sense oxygen. I mean, what can be more important, right, for living organisms? And you move from the very basic, even evolutionary, as you show at the end of the talk, to the translational aspects. And I think this is wonderful. But the question has to do with, there is this push for translational medicine that we see constantly, having to justify the justification in our grants is huge, really. And unfortunately, there is apparently an apparent dichotomy between basic sciences and translational medicine sometimes. Sometimes people interpret this as one versus the other. And of course, I don't think like that. And you are an example of that the dichotomy doesn't exist. But I just want to hear your thoughts about it.
We started out with a very defined question, right? What regulates the EPO gene? And then that led to a discovery that had very broad implications. But that was, that was not part of the grand plan, right? We asked a simple question, and then we got a very remarkable answer. And it's that serendipity that you don't know where the research is going to lead, and you can't really focus and say, I want to go from here to here, because it's never a straight line. And so you need basic research, because basic research answers basic fundamental questions. And then the result of those, the answers to those questions will have therapeutic implications by their nature, because if you're affecting something that's fundamental, of course, it's going to affect human biology and human disease pathophysiology.
Thank you so much for your meeting with us, your talk today. And we still have a little more of work for you. From 3 to 4, there is a Meet the Nobel Laureate session, and people, trainees, and all of us, really, we have the opportunity to continue learning from you. And again, on behalf of the Council on Hypertension, the American Heart, thank you so much for your time and for your efforts in doing this for us. We are indebted to you.
Thank you, Dr. Gomez. My privilege and pleasure. Thank you very much.